So today we are going to talk about telomeres and the enzyme telomerase. So first is, what are telomeres? So telomeres are basically the ends of the chromosomes, or you could just say because DNA is one big molecule, the very end or the ends are going to be telomeres. Now, telomeres are actually tandem repeats, which mean that repeat over and over again. These include the most famous is the AGGGTT. So these are repeated over and over again, and that's why they are called as the tandem repeats. Now, the thing is, another important thing to know is that their length at the end, so this is the end of right here, this is the end of the DNA molecule, and this essentially are this is the telomere. And this telomere, important thing to note is that there are two particular strands. So the end, so we will now look at the ends, the 3' end and the 5' end. So this strand, the above strand, is the 5' to 3' strand, and below it's 3' to 5'. Right? So because of this, you will notice that this is a G-rich strand. So this strand is G-rich, and this strand is C-rich. The thing to notice here is that G-rich strand is longer than the C-rich strand. So that is a bit of a structure of a telomere. Now, we have to see, it's important to know that they shorten with each mitosis. So they shorten with each mitosis. Now, why does this happen? Because of lack of filling of gaps. Now, I'll tell you what that means. I'm going to zoom it in here. The thing is that we have this long strand of DNA, right? And it is replicating in this direction or whatever you want. It's replicated. Now, after replication, we have is DNA repair, right? During DNA repair, what actually happens is that the bases are excised and then they're removed, and then DNA ligase and DNA pol 2 comes and they fill in the gaps. But what actually happens is that the very end, this DNA repair mechanism, because it's includes enzymes, these all enzymes, they are they're just like a car, right? When you have a car, just take an example. If you have a car right here and this is a road, the car will stop somewhere here. It might not complete its journey, right? Because its energy is out, its petrol or its engine oil is out, whatever you want to say it, right? The same case is with the DNA repair enzymes. They cannot reach the very end. Because they cannot repair the very ends of the DNA, they are lost. So telomeres are actually protective. They're altruistic in nature. They will, actually sacrifice themselves so that important information of DNA is not lost. So at the very end, there is no important information. It's just introns, or you could say tandem repeats of information at the ends. So that's what telomeres do. That's why we call them mitotic clocks. That's why they are the mitotic clocks. By looking at the telomeres, we can see how many mitoses one cell has done. Now, some cells, we move on now. Now, some cells have some enzymes called as the telomerase enzyme. Now we're going to talk about what are telomerase enzymes. Now, telomerase enzymes are going to lengthen, they're going to increase the size of telomeres. So they increase telomeres. Now, how does they do that? Telomerase enzymes are actually reverse transcriptase enzymes. So I write it here for you. Reverse transcriptase. Now, these reverse transcriptase enzymes are essentially the same class of enzymes as you have in the HIV AIDS virus. That's the same. Now, how does it work? Now, we see this is our normal telomerase strands. So, the telomerase enzyme, it is this one right here. I made it. This telomerase enzyme is multi-subunited, and it has, sites. So it has the enzyme, this is the enzyme right here, and it also has an RNA primer. So this is the RNA primer attached to the telomerase enzyme. And that RNA primer reads 3' to 5'. So you see U, C, 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 A, A. So U means that this is from the RNA primer. Now this is going to give you an RNA primer. Now, as we have this RNA primer with us, now DNA pol alpha can come and increase the size in this direction. It is going to replicate in this direction, and that is going to lengthen the 5' end strand, right? And in the same way, the 3' end strand is also lengthened from the same enzyme. That's how telomeres are lengthened. And these, types of enzymes you find in cancer cells. So many different cancer cells have telomerase enzyme that can increase the telomeres, and essentially the cells won't age, right? So with regards to aging, telomeres, what others think that we need to know is that we can, telomeres are cause of primary aging or senescence. And we can also use these to track certain diseases like progerias, etc. Right? So that is it for telomeres and telomerase enzyme. Thank you so much.